Good morning, everyone. Thank you for joining us this morning, being the first session in the morning. I would like to welcome all of you here as we kick off day 2 in meeting on the Mesa. So I think, as many of us here can agree, the cell and gene therapy has reached significant milestones over the past decade. There are many firsts. It's hard to imagine that the first CAR T therapy was only approved like 6 years ago. And we have the first allogeneic therapy approved less than a year ago, and also, more recently, the first approved cellular therapy to treat type 1 diabetes only a couple months ago. So very exciting to be here today. And over this period, I think we have learned a lot. Not only in terms of clinical and patient treatment management, but also in terms of understanding our manufacturing processes, defining critical attributes, supply chain, and logistics management. So my name is Mayasari Lim, and I am the Associate Director for Life Science Business and Product Supply at FUJIFILM Irvine Scientific. We are a global life science and medical device company specializing in the manufacture of cell culture media, reagents, and assisted reproductive technology products. Our headquarters is right here in Santa Ana, Orange County, which is only an hour away. We also have global manufacturing sites in Tokyo, Japan, and also more recently in Tilburg, Netherlands. So today I'm very, very pleased to be joined by an esteemed panel of speakers who bring deep expertise and diverse experience in the biopharma, biologics, cell therapy manufacturing, representing critical raw material suppliers and service providers in the cell and gene therapy space. Our speakers understand the complex challenges in cell therapy product manufacturing and development. So they're here to address how a close partnership with your suppliers can help accelerate your development process from research to commercialization in the shortest possible time. So let me first introduce each speaker to you. First, I would like to introduce Dave Smith, President and Chief Commercial Officer of Akron Biotech. Dave has more than 20 years of experience in the cell and gene therapy industry as a manufacturer, ancillary material provider, and cell therapy product developer across all regions of the world. He is using his background in manufacturing to bring raw materials that are production and regulatory friendly to the market, and we're very happy to have him here with us today as one of our panel speakers. So I'll let Dave tell us a little bit more about Akron Biotech.
Akron Bio is actually 2 different divisions. One of them is actually a product division where we bring ancillary products for the cell therapy market, growth factors, cytokines. And what we did over the last number of years, we've been here since 2007, but we've changed how we were doing things. We made it much more manufacturing friendly. Mm-hmm. As you know, the toughest thing for cell therapy manufacturing techs is prepping media early in the morning, and it takes up time. Now we're moving much more to ready-to-use liquid cytokines, products in bags so we can weld and go. On the service side of the business, we have a plasmid facility, which we were able to not only do plasmids, but we've just completed our second nuclease manufacturing program. So we're not only taking our E. coli base and not only doing plasmids, which at a very high level, but we're actually taking it over and also doing nucleases. Again, in support, ancillary materials in support of both the cell and gene market.
Thank you. Thank you, Dave. Next with us, we have Dr. Tim Kelly, Chief Executive Officer of RoosterBio. Tim joined RoosterBio in October 2021 as the CEO. And previously, he served as president of manufacturing at AskBio, which was acquired by Bayer in October 2020. And for 15 years prior, he was with KBI Biopharma, an industry-leading contract development and manufacturing organization, where he ultimately served as president and CEO. So Tim started his career with Diosynth Biotechnology, and as one of our esteemed speakers today, he brings a wealth of experience in development and manufacturing services for over 325 biologics throughout his career in all stages of product development and commercialization. He has also supported numerous successful FDA and international regulatory inspections throughout his career. So thank you, Tim, for joining us today. Please tell us a little bit more about RoosterBio.
Oh, thanks, Maya. Thanks for inviting me to participate here today. So RoosterBio was founded 10 years ago with the mission to industrialize the supply chain for cellular supply chain for advanced therapies and deliver really scalable manufacturing solutions, to the advanced therapy space. Right, some of the key challenges, that have we think kind of held back, the development and really the impact of these products. So we're really focused, so far focused highly on mesenchymal stem cells and exosomes derived from MSCs. So we have off-the-shelf GMP grade cellular starting materials, multiple tissue types, multiple donors, and then we pair that with highly engineered media that's designed to work in bioreactors in a sort of no media exchange, type of bioprocess, so that we can actually generate, clinically and commercially relevant quantities of cells and exosomes. So really the key, with our business model is it's, taking the cell, making that highly accessible. You can purchase GMP grade off the shelf, pair that with, the media that allows you to, scale those processes and deliver a cost of goods that makes sense for patients and providers. And then we've built the bioprocess around that, so that it's really a manufacturing system that can be readily implemented, for our customers in their own labs and manufacturing facilities.
Thank you, Tim. Thank you. Okay, last but not least, we have Dr. Haro Hartounian, founder, president, and CEO of BioCentriq, a thriving cell and gene therapy CDMO. Haro has played a visionary role in establishing BioCentriq, introducing this unique facility located in Newark, New Jersey. Haro has almost 30 years of experience in the biotech industry, working with both large and mid-sized biopharma companies. He's recognized as a dynamic leader and organization builder, consistently achieving remarkable performance and driving growth throughout his career. So Haro's impressive career included pivotal roles in successful launches of 2 FDA-approved products, Depocyte and Depomorphin, and his expertise extends to serving CEO and board member of 2 publicly traded companies and 3 privately held biopharmaceutical firms. So Haro, thank you for joining us. I'll let you introduce a little bit about BioCentriq.
Thank you so much for the introduction. BioCentriq is a global cell therapy CDMO. We launched BioCentriq officially in 2019, and in a short period of time, the BioCentriq was acquired by Green Cross in South Korea. We're a global CDMO. We have capabilities, clean rooms, in New Jersey, as well as 6 clean rooms in South Korea, Seoul. So we can cover all North America as well as the Asia market. We are a unique CDMO such that we focus on early stage biotech companies that they don't have the infrastructure to develop their product or produce their clinical materials for their clinical trials. So we have ongoing partnership with those companies. As a matter of fact, in a short period of time, it was a major accomplishment for our CDMO that we produce material that those materials are being dosed in 11 patients now, which is a major accomplishment for us. The team is close to 75 people, that we have people, chemical engineers, process engineers, scientists. So I welcome whoever has a project that's looking for a solid CDMO, come and utilize BioCentriq and use us for your product development. Thank you.
Thank you, Haro. Great, so now that we know a little bit more about our speakers and also the companies they represent, I wanted to kick off with talking about the selection of critical raw materials. Since all of you have worked closely with numerous cell and gene therapy manufacturers, what advice can you give to the audience today in regards to critical factors that one should be thinking about when it comes to selecting raw materials, whether it's for cell culture media or other reagents that are used in cell and gene therapy product manufacturing? So maybe Dave, do you want to start first?
Sure. Thank you. My first thing I would say is it's about the supplier and about being prepared for what you are going to do for the future. You need to think about your end point. Too many times we make decisions around suppliers of critical raw materials early in our process and haven't thought about, does this carry me through if I'm successful? making changes in these suppliers is difficult, and often these materials cannot be reproduced at larger scale. So you may have to choose a slightly different supplier, a different raw material, do it early, make those decisions when early on. The other thing to consider very quickly is, does that raw material, does that critical raw material get me to other countries outside of the US? The regulations around Japan are a little more challenging. Europe has some high challenges, but at this point we are working very hard at our group, which is for the FDA we do ECTD filing. So all of our raw materials have a filing at the FDA. You simply go in and you get a letter of authorization and you go forward. The same thing we've done now even with the PMDA. There are a number of our critical raw materials that we've gone to the PMDA, did full filings on them, and made sure that our customers can take it from here into a clinical trial in Europe, into a clinical trial in Asia, and without making changes. These thoughts very early on in your process will make you successful.
Very good point. I 100% agree with you, Dave. I think the regulatory challenges is definitely a challenge for all of us. Tim, do you want to add?
Yeah, no, so certainly echo everything that Dave said, spot on. I'd say a couple of things I'd add to that are, you got to you kind of mentioned sort of starting with the end in mind. And I think, with advanced therapies, we also have to recognize that in many cases, these products are not as highly processed as other biologics, right? So that even amps up the criticality around the raw materials in your cell culture media, for example. So certainly taking, a really, sort of thoughtful approach, a risk-based approach around selecting raw materials. It's really not practical to develop your process with GMP-grade materials. So really understanding, having a supplier that provides research grade, but also supplies GMP grade, and then knowing the differences between those two. Ideally, the same critical raw materials are going into the research grade as the GMP grade, so that when you're ready to move to GMP, you're not having to re-optimize your process, you're not getting surprised by process performance. So I think really understanding how your suppliers sort of have thought about their own supply chain around both their non-GMP and GMP grade materials, I think is important. That's something that we definitely strive to do, right? Our kind of development grade media is exactly the same as our GMP, all the same raw materials. It's just manufactured outside the clean room, right? So it really does enable kind of that seamless transition to clinical manufacturing when our customers are ready.
Thank you, Tim. Haro, anything to add?
I can speak on the CDMO's perspective,
Yes,
because
please.
we help the clients, they come and work with us. I think the first thing that I recommend the companies to identify the critical raw material that they need in their processing. And then also choose a supplier that is viable. There are a lot of suppliers that are out there that they are there one day and they're not there. You have to have a secondary supplier too, and making sure that what they're offering you is a lot variability. Get them involved from the beginning of the process, and make sure that they know what exactly you're looking for. We will talk about the regulatory requirements, but I think that's another aspect of making sure that they comply with GMP manufacturing, they have all the CFAs, and even certificate of compliance and all those things in place. Those are things that you have to put a check mark and say, okay, check, and make sure that supplier is a viable supplier for your process.
Yeah, absolutely. And I agree with you, Haro, like thinking as early as possible, it's important, and having those conversations with the supplier early is also important, because if you start with not selecting the right raw materials or suppliers in the very beginning, then you're going to end up, backtracking or changing supplies. It's just really a nightmare. So definitely the early stage is very, very important consideration. I think from one more thing I just want to add, because I work with a lot of customers who come to us with custom cell culture media manufacturing, and I think definitely that's also one of the key things, is that the earlier we have that opportunity to work with our clients on developing or optimizing their cell culture media, then the better success they have with putting them in the right path. So definitely.
May I add one?
Yes, please.
I think also one comes to mind which is very important. A lot of this group will work with autologous products. it's very important for you to test multiple batches of whatever raw material you're using. Quite often, it's great. I will test the batch, it works really well, we can go forward. Always test, if you're going to use it, you test multiple batches. It's very common in the reagent market to have fairly high variability lot to lot. So what you can do is you have a patient who can be very, very variable as you're doing your processing. You have raw materials that are very variable, and you get really good and you get really bad and you don't know what is driving it. So you look for companies who can do 3 lots consistently, give you that yield, give you that performance that you're looking for, because those are the groups that, for one thing, they have 3 lots, which is good, and they have it available for you, so you know you're not going to go on back order, and you know they have consistency lot to lot. You should actually ask for their lot trending data. They should be able to provide you that to show you that you are getting the same product all the time.
Thank you so much, Dave. I think you're helping me segue to the next question. So I think leading on to that, you know, after one have selected like the critical raw materials and components, what are some of the quality attributes, right, that one should be considering in terms of minimizing risk, lot-to-lot consistency, and then just reducing variability overall throughout the manufacturing process? So maybe Haro, we'll start with you first.
Sure. I know that I'm sure that you guys are aware that companies, the FDA now is requiring companies to do an independent ID test, meaning that you can't just rely on the CFA, and you have to have an independent firm to test all the raw material, making sure that they meet the specification you're looking for. As far as the quality attribute, of course, you have to look at again, I always go lot-to-lot variability and making sure that, you know, that you get the same product every time. For example, if you're working on the media, the sterility of the media that you use, and they sell you serum versus serum media. Those are the things that we always recommend people when they do cell culture. And they play a very important role in the quality of the material, the growth of your cells that you do in your bioreactor.
Right. Yeah. Tim?
Yeah, so I guess a couple things, just thinking purely on the kind of quality side, you know, I think one of the things that can be tricky that's important to understand how your suppliers manage are change notifications. Right. You know, and really, you know, having the right quality agreements in place to ensure that, you know, you're aware of process changes. You know, major, minor can get really dicey, right? You know, it's the eye of the beholder territory, right? So really making sure that you're fully aware of how your suppliers are managing change, because again, in these types of critical raw materials for biologics, this is not simple stuff. You know, it's not just salts and you know buffer species and whatnot. And then I guess the other way I look at this question is around the design of the products. You know, how those products are designed to use, because when you think about the risk of variability and you know, kind of process performance, you know, how the products are designed to be used makes a big difference. You know, for example, again, in the cell culture media space, if you're working with adherent cells, most of the media that you're using require media exchanges. So every time you do a media exchange, you're adding risk, you know, to your process, right? There's just sterility risk. Every time you manipulate your cells, you're adding risk, and you know, you just have process performance risk every time you change media. So thinking about and trying to find products that are designed to minimize process risk, just in how they're formulated, how they're designed to work, I think is an important part of managing that quality profile. You know, certainly that's a big part of how we try to design our products, is to minimize risk and optimize consistency, right? So you get the same number of cells at the same population doubling every time, you know, through the passaging of the cells, you know, and of course, you know, with our system, you know, we're avoiding media exchanges entirely, which, you know, we think adds not only a lot of, you know, cost of goods value, but, you know, significant risk mitigation, you know, for our customers.
We're just going to add something about that. Then one of the things that you can address is to put that quality agreement that you put together with your supplier and making sure that they...
No, it's essential, yeah.
That's correct. So they follow all the change control and everything else that they happen. So It's very important. We always do that.
Yeah, that can't be the afterthought, right?
Right.
You know, quality has to be at the front of the selection process, right? The quality agreement should come before the supply agreement. You know, before everything. The audit needs to happen. You know, before you make these decisions, because you know what would people say, and you know what actually is happening. Sometimes aren't consistent.
I would add, along with the same with the QAA, but some of these are just your regular raw material suppliers. You know, you have critical raw materials. You may think that, why do I need a QAA? Why do I need an agreement at all? Because I'm just buying, a media off the shelf from Sartorius, and you know what? If it's on your critical raw material list, face to face at the facility, you want to see where they're making it. Yeah, it's expensive, but it's worthwhile. A QAA, very worthwhile, and making sure that you can look at a few of the batch records. Make sure you go in and take the time to do, even it's a half-day audit on your suppliers. It's perfect, because these are the people you're going to be with for the next 5, 10 years. And so you want to make sure that they look viable, they have a track record, they can show you trending data on your material. These are the things that will save you a lot of manufacturing headache, because there's, coming from 18 years of contract manufacturing, trying to figure out what problem you all once are getting in your cell culture, because you can't figure out what raw material changed. It's troubling, it's a nightmare, and it takes you forever. So being able to pick those very early will save you, days and weeks of trying to figure out a troubleshoot along the way.
Yeah, definitely. Thank you, Dave, for commenting on that. I think earlier you touched on the regulatory aspect, so I want to go back to that and talk about, what are some of the things that you think one should be asking, right? There are suppliers as far as getting more support and ensuring that they have the best path forward for success for their regulatory filing. So what advice would you give to our audience here?
I think you get the middle. You're first.
Want me to start out? Right? Easy. It's good to go first. No one steals your comments. So I think there's some obvious things here, right? And I think it starts with, if you're talking about critical raw materials, going into an advanced therapy process, then having those be supported, at least here in the U.S., by master files, is kind of table stakes, right? I think that's something that you should expect, and that certainly really accelerates and de-risks your regulatory submissions, right? If you can cross-reference a master file, to take care of, again, for these advanced therapies, cells and media, are a huge chunk of the CMC. So if you can cover that, through an LOA to a master file, not only, again, are you saving yourself a lot of work, but you're really, kind of de-risking the whole process, because FDA has already reviewed those master files for other LOAs, and you can have some assurance there. I think that beyond the kind of the basics there, I think it's having a strong relationship with your suppliers for regulatory support, because obviously the master file doesn't do you a lot of good outside the U.S. So understanding how your suppliers are able to support you for international regulatory filings, how willing they're going to be to put the effort in, to supplying those dossier support packages, because that'll be necessary. And again, understanding how sort of they've done their homework. Again, we kind of touched on different expectations, different requirements for different regulatory jurisdictions. So kind of, again, when you're talking about raw materials that include biologics, which again in many cases, for advanced therapies, these critical raw materials are biological in origin. It's great to be able to use recombinants, but that's not always feasible, either from availability or from a performance point of view. So understanding exactly how those materials have been sort of qualified by your suppliers, what type of screening and risk assessment and testing they've done. Again, how does that fit in not only with U.S. expectations, but with international expectations, particularly PMDA, as Dave mentioned, because that's definitely a different ballgame.
Yeah.
I think the first thing that we always do is to do the audit. If we select a supplier, we do the audit and making sure that they comply with the regulatory requirements. I think you have to have that. I agree with Tim that you have to build that relationship with your supplier.
Yeah.
Because I think if you don't feel comfortable with your supplier, then you'll always second guess. I.e., for example, do they have the right change control in their system? If they change a process, do they let you know? Because you need to know that and use your filings with the FDA. So that's very important to make sure, are they trained and the people comply with the GMP manufacturing? All those things that you do to your company, you should expect them to do the same thing, because at the end of the day, you have to file with FDA, and as I agree that the master file is not sufficient. So they have to have the infrastructure to support the FDA filings.
Yeah, definitely.
Agree with many things. I also like to see, are they up to date with their filings? One of the things is 2-3 years ago, the FDA started accepting electronic filings for, so ECTD filings are pretty straightforward. It means that there's systems and people understand that. It kind of forces you to update it once a year, because sometimes, came from companies in the past, you did your master file and never looked at it for 5 or 6 years and did not actually update it. These are the things you want to see. Is their quality group up to date? Do they actually understand what they're doing? The other thing is, see how much they've stretched out. 2 years ago, I had done a stint in Japan for a few years, and I became very good understanding of the PMDA and the raw materials and requirements. You can go to Japan and get a letter of authorization from the PMDA that says we will accept this raw material into your product going forward. And so when you have those kind of assurances that it will work in the US, you can use it in Europe, and oh, by the way, the Asian market is bigger than the European market right now, that you can take it into Korea, Japan, Singapore, Taiwan, and sometimes China. That one's a tough one for everyone. But those are the things that will get you there. And one other thing that I kind of talked about, lot trending. Look what people's specs are. You will often see, like for cytokines, you will see people, they have a variability somewhere, it will be at least 22 MIU. That 22 MIU could be anywhere. Some of these people have fill criteria between 19 and 40. And so you have these big giant variabilities in their filling process because they don't want to take the chance. They make sure they put enough product in. Right. But you don't know if it's 20 this time and 40 next time, and it's a big, because their specs are designed to be very open because they don't ever want to fail a lot. So again, the lot trending, it can be very, very tricky, especially as you're doing batch to batch variability in your own institute. Like, why didn't this work for me this time?
Yeah, very, very good point. Dave, I think that's definitely an important thing to consider, is really looking at the details. The devil is in the details, right? So, yeah, I think with international waters, definitely it's very challenging, at least from my experience. I think that, having the right SME, so to speak, right, understanding different regulatory requirements for each country, our region, is absolutely critical to ensure success. So that's something that we have also learned.
Can I just add something? Maybe I want to ask my colleagues to comment on that, because the secondary supplier, I mean,
Yes.
typically, is FDA requires to have a secondary supplier or not? Is this a requirement? No?
It's not a requirement, no. Certainly advisable,
advisable,
but,
yeah.
not always. Again, when you're talking about biologics, it gets tricky, right, in terms of actually finding a secondary supplier that gives you not only all these quality and regulatory attributes we've been talking about, but just performance, right?
Yeah.
Right.
So yeah, definitely advisable, but I think sometimes it's a high bar, to reach.
It is, yeah.
I would say it's a very high bar. Everyone likes, in the farm, as we work with a lot of farmers, it's very hard for them because they have criteria that says they have to have a backup supplier often in their, especially as they're getting commercial.
Yep.
It's very, very difficult for them to find one and get the comparability data.
That's
So-
what I'm, yeah, that's
the
what I'm,
challenge.
but most of them, they don't, is that right? They don't have a secondary supplier.
It depends on the component, right?
Component, yeah.
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 But good question. So I think segue to the next question that I have for you guys. It's, you know, I think we've seen a lot of progress and traction in the field already. We want to start thinking about going beyond phase 1, right? So what are some advice you can give as far as, you know, scaling up, supply chain, things like that? So Haro, do you want to start?
Sure. I think any cell culture and bioreactor, you always use the QBD, quality by design, and identify your CQAs and CPPs and try to actually shoot to make sure that you get all the parameters that you're looking for. In my experience, I think on the processing side, of course, you look at the pH, look at all the stuff that you look for. On the biological side, you look for cell growth, cell viability that you envision. So scale up. Typically what we do is, and I was talking to Tim about it, that we use amber, for example, to optimize our cell culture, and then is it always linear to go from an amber to a 50 liter or 200 liter or 2000 liter bioreactor? It depends on the cell. Tim educated me today that, but it's not always linear.
Right.
And it's challenging. And I was just mentioning about a company that we were talking that they're doing NK cells, right? And they are optimized the process in 50 liter, but they're having challenges to scale it up to 200 liter. And it's not always scale up a linear process. So those are things that you have to initially identify. Again, your CQA and critical process parameters, and start working to optimize and make sure that you have a successful scale up.
Absolutely. Dave, do you want to say?
So scale up is, I think you guys can also agree, this is autologous allogeneic. You still, in some ways, yes, everyone talks about we should be moving over to allogeneic everything, and we should be putting it in bioreactors, and it fits the pharmaceutical model. And however, it's not always possible.
Right.
So we have to also think about the scale up on the autologous side, which is what we're talking today. This is where you start talking, thinking about how do you simplify the media processes or the manufacturing process to the very basic level, and try to take out places for error. Because you have, if you look at a basic CAR T process, the number of technician touches on that is simply incredible. And so you start doing things like, we've been working on weldable cytokine, weldable media, weldable proteins. So that basically, I don't want a technician to ever reconstitute a cytokine, sterile filter it, inject it into a bag to prep my media. It takes up time in your clean room, and it's a place of risk. You want liquid cytokines, you simply hook them up and go. You want your either HSA or AB, whatever you're using, you want it in a bag. You want it just to weld it to your system. As we go closer and closer to automation, we're going to have to take out every place that technicians touch, because that is where we add the risks. That's where we add, a deviation adds hours and hours of labor, as we've all been through that. The deviation and investigation, and you just took your quality group offline for 3 days while they're doing an investigation. We need to get all those touch points out that we possibly can. So that's where I would think we close it up, we make it work, with all the automation platforms coming forward.
Yeah, so I'll kind of focus more on allogeneic. That's certainly where we focus, and where I hope our industry continues to point. Obviously we've got some technical challenges yet to overcome, but upstream, I think others have touched on it. I think kind of getting into bioreactors. It's possible to do this I think with any cell type, with the right engineering, right, so we're growing. Our MSC process is a microcarrier-based, 3D bioreactor process. It can be scaled. Can we get to the thousands of liters? I think that's more a function of, for MSCs, the PDLs that are available, and hopefully, moving into immortalized, cells that get us to greater scale, because that's what's going to be required, right? If we're able to develop these therapies to treat huge indications like stroke, we're not going to be able to do that in 2D systems, right? We have to, move into bioreactors, and we have to have raw materials that support that. But I also want to touch on downstream, because in a lot of cases with these advanced therapies, the downstream processes, to the extent that there is much of a downstream, is not at all scalable, right? It's dependent on things like centrifugation or even ultracentrifugation that, you see these, clean rooms that people are using to make AAVs still that are just wall-to-wall centrifuges to do density gradient ultracentrifugation, right? That's why these are so expensive, right? We have to have, different ways to process these materials using scalable downstream unit operations, right? Take a page from the therapeutic protein book and let's figure out how to engineer these downstream processes with, TFF, with chromatography, with unit ops that are amenable to scaling, because a lot of the operations that are being used today in advanced therapy for downstream are just not scalable, right? You have to, it looks more like an autologous process in the sense that you just scale out, right? You just add more centrifuges, to process. And you can do it to a certain extent, but it's ultimately going to limit the impact of these therapies, if we can't do this in a more scalable and cost-effective way.
Yeah. I think it's interesting, Tim, because if you think about it, we've been talking about scaling up bioreactors since 2010, 2011. It's really not, for all the talk and all the little steps we made, we really haven't found that path yet, I don't think. It's something that we all, at least I think about it, like we need to get all these to the next step. And I think the CAR-T business has become, was a boon to the industry, but it took the focus a little bit away from the allogeneic process, sure. And all the automation, all the everything moved to kind of scale out, as you said. And now we're going to see more automation platforms. There's been lots of cash spent on it. We need to come back, and as you said, the downstream on the bioreactors is still lots of challenges.
Absolutely.
Yeah. I think the autologous side, cell expansion, there are a lot of automation going on. Closed systems are being developed. I think that's technologies catching up to address the concern that you have, the operator, and the time of the processing. So I think the technology is getting there in the autologous side. The allogeneic side, of course, there are many other challenges in addition to the scale-up. Just the cell viability and all the genetically modification of the allogeneic cells and stuff like that we should address them.
So hopefully next time round, next year, we can talk more allogeneic scale-up.
Work is, they're doing it, so we're getting there.
I want to leave time for the audience to ask some questions. So love to hear from the audience here if you have any questions for the panels, and also maybe if you want to share some of the pain points that you have. So feel free to ask questions. So anybody like to start? Yes, please. Do we have the mic that we can? The small box.
Chat, introduce yourself.
Thank you.
Got a plant.
Hi, Chad Silva from BioCentriq. Interesting discussion. Thanks for being here this morning. We didn't touch much on cost of goods, right? So if you're thinking about a commercial program in the next 10 years, right? How do you factor that into supply selection, optimization of that, scale up you talked about? At what point and how does that become a consideration?
Okay.
Sorry, what was the?
The question was related to cost of goods.
Oh, cost of goods, yeah. So I'm happy to start. Yeah, I mean, I think it kind of, to me, it's all interwoven, right? I think, a scalable process kind of helps your cost of goods, right? Things like, again, choosing raw materials that enable the most efficient process, that drives down your cost of goods, using less media, using, less, buffers and reagents, so kind of getting to this, kind of next generation, processing, thinking about perfusion processes, if that's relevant, to sort of, extend, sort of the production window for various therapies. But then ultimately, again, this is, I think, the challenge that we have, right, with whether it's autologous or, again, some of the non-scalable, downstream processes. I think that's what's contributing to the cost of goods. So I think those are the challenges that need to be solved in order for, these therapies to become accessible, right, and not cost $500,000 a dose, which is, I mean, it's an amazing thing that we have therapies that can do what they're doing, in terms of remission rates. But as more and more of those come to market, I think the challenge is only going to become greater, right, for how this actually gets paid for. Right now, when there's only a handful of gene therapies and gene-modified cell therapies and other types of products out there, I think we're kind of at the tip of the iceberg for the challenges that we're going to face with how this gets done. So certainly the move from autologous to allogeneic, I think, is critical for that. But even within allogeneic, it's not enough just to be allogeneic. I think, again, it has to be an efficient process that gets high productivity with relatively low cost in terms of the inputs, and manufacturing time is a big part of it, right? A lot of these therapies, you're in the clean room for a month, to make a handful of doses, so that you're never going to get attractive cost of goods, especially not in the U.S. With that type of process. So we have to address scalability, in order to answer the COGS question.
I think that to just add, sometimes cost of goods are there. You can't change it, right? I always think that innovation plays a very important role to reduce your cost of goods when you plan to scale up and commercialize, i.e., for example, non-viral vector transfection, using automated systems. I'm talking about the autologous cell therapy, closed system, less labor cost, all those things. Innovation will play a very important role to reduce your cost, and using innovative technologies in your processing.
So one of the things I would think about is there's a number of really good models. And I know we have to model this out very well, because in the end, we're going to have to kind of stand up to that number and tell investors this is what it is, and this is what we're going to get to, and this is how we're going to get to the next step. And some of these we're going to have to think about changing the paradigms all completely. And my favorite one is, you think about today, we're treating maybe 15 to 20,000 CAR T patients a year in 5 different facilities, and they max out about 5,000 patients. They're maxed. What happens when we get the product, gets over the wall, we get an INK that gets into solid tumors? Now you're talking about 200,000 lung, 100,000 ovarian, prostate. We have to just go blow it up and almost rethink about it, because if you're going to use a base of an IPS to get to an INK, slow grower, think about autologous CAR T, or allogeneic CAR T, those cells don't grow particularly fast. Their doubling time isn't particularly fast. We're going to have to think about how do we change the growth parameters. Can we get it further downstream so that we do a converted NK cell that comes from an IPA cell and then expand that? Like I said, I think we're at a point where we really have to put that mindset of what happens when somebody actually gets into solid tumors. And it is going to be you're going to have to put the best minds together to really work through this, because I don't think there's any solution today to get to the numbers that we'd have to get to.
No, I think it's a good point. I think the way that we try to think about this is we sort of try to envision the type of process that's required to deliver of the therapy, right? So it's kind of a process-first approach, and then, okay, what are the products that are not available on the market today that would be necessary to drive that process, right? To deliver on that process. So I think thinking about, again, sort of cell therapy approaches to solid tumors, looking at the number of patients, trying to understand, what does that process need to look like in order for, us to deliver on that. And then what's missing, right? What's not available today to deliver on that type of process. And that's where companies like, frankly, ourselves, right, the tools providers of the world, I think, have the role to play, right, is to help the pharmaceutical developers figure that stuff out. We need to invest more in, kind of how to generate these, scalable, manufacturing processes for advanced therapies. I think too often we're happy to just sell lots of media, sell lots of cell stacks and lots of prodigies and lots of bioreactor bags and so forth, without trying to really tackle the problem around how do we make these processes scalable and viable for kind of that type of end user. I think it's a great point in terms of how are we going to address because the science is going to get there, right? We've seen how far we've come just in the last 7 years, with both vector gene therapies and CAR T. We are going to get there with allogeneic. We are going to get there with solid tumors, but we're not ready. We don't have the tools to deliver on those opportunities yet.
Yeah, and I like the same. Sorry, I mean, think about if we make small changes. Like, so we've seen people who are going to like 2-day, 3-day CAR T processes, right? You just kick the problem down the road in the sense that we can't test. I mean, then becomes testing and quality. You cannot test 100,000 CAR T samples in a year. I mean, there's not the system right now. I don't know, you guys. The biggest backlog. It's a guess every time I know. Every time I have to send testing out, what day it's coming back is a big guess most of the time. It's a bit of a challenge, but think about if we flood the system with 100,000 samples. So we need to automate. We need to automate that part of the process too. And there's a lot of people working on electronic batch records and release by exception. That's probably a little further, but for me, QC is the next wild card.
Ramis Bistras, Darasis Therapeutics, we're developing gene therapy for glaucoma. So a question I have coming back to due diligence and selection of CDMOs. From your experience, and I want to connect that the pricing also. From your experience, what are the top 3 kind of due diligence points you want to look at CDMOs, and how do you evaluate that versus pricing? Let's say lower pricing. What could you compromise in due diligence?
I can answer that question since I'm a CDMO representative. Very good question. Very good question. I've been in your shoes and I've been checking CDMOs and identifying the good ones. The first thing is the track record and making sure that that CDMO has developed or produced material that's being used in clinical trials and being put in patients. That's the first question you ask. Have you done this before? That tells you about their quality system, the infrastructure, all the things that you're looking for, your filings, right? So that's number 1. Number 2 is the team that they have. Because, of course, the SMEs that they have, specifically for your therapeutics you're working on, if you're working on specific cell lines, do they have the people there that they can help you to optimize your process? Do they have the right MSAT team? Because you do a tech transfer. The companies, they come to us, especially the early stage companies, they think that they're ready to go and sell their product, right? In reality, they're not. So you need to pick a CDMO that will challenge you and ask questions. That there's an issue here, do you agree with that? And collaboration, are they collaborative? They have a collaborative team that they can help you to develop your product, do a successful tech transfer, do they have a good MSAT team, how many pilot runs they have done, engineering runs, all the stuff that you require to get to the clinic. So I think those are 3 factors and also stability of the CDMO. Because the CDMOs, after this COVID, they grew like a mushroom. Everybody started building CDMOs. and now all these clean rooms are empty. So are they going to be around? Is this CDMO going to be around when I'm ready to commercialize my product 3 years later? BioCentriq is very stable. You know why? Because we have a multi-billion-dollar company as a major shareholder from South Korea. So if you have a product, come to us, please.
I gotta go. So I was employee one with Cambrix, Lonza. So I built the Lonza CMO world. So yeah, you and I have about the same. But since I've left there, retired, and went on to other things, I've had to go back and pick CMOs. And when it comes right down to it, a couple of things. One is, you said sustainability. Are they going to be there? Do they have quality systems and really can work with it? And for me, you got to know that you can work with them. Because you know what? There is never a time in cell therapy there is not going to be a screw up. Right. It is a natural thing. You know, we aren't making sterile products. We are making low bioburden products. There will be a contamination. There will be a deviation. You have to know that no one loses their mind. Because if you all start locking up and going, oh no, this is yours, I don't have to pay for this, it will go bad quickly. And so you may end up choosing somebody who is maybe a little smaller, who you can work with. But it's especially in cell therapy, not going to a machine is probably a good thing, especially if you're in early trials.
I totally agree with you, because I'm not going to mention the name of the CDMO. My third company I founded, a biotechnology company, and it was a virtual company we raised. It's a very good question the gentleman asked, and it's personal experience. We raised seed funds, right? It was a virtual company, and the biggest mistake I made, I went to one of the biggest CDMOs to help me develop my product and make the clinical material. Guess what? They didn't pay attention to me at all. They put their 4th-tier, 5th-tier scientists, engineers on my project. It took us 2 years. They used every single dollar I had in my bank because we had to shut down the company. So coming to a smaller CDMO, you will get more attention and better product.
Yeah. Ask them what their turnover rate is. That's the other one.
That's correct.
Right, so none of you guys have talked about cost yet, which was specifically part of the. So, I mean, there is an element of you get what you pay for, right, and everything. And I think the only thing I'll add to this, because you guys covered it well, is the relationship is absolutely the most important thing. And you'll get this through all of these, your quality audit, your discussions around, your supply agreement and so forth. And then references, obviously, you need to talk to customers, of that CMO, because, when it hits the fan is when it matters, right? How is this, going to work with you as a partner? The one thing I would say, the one, I guess, request I would make, please don't turn this into a procurement, process, right? Don't let your procurement groups make decisions about your CMO partner or about critical raw material suppliers, right? These are too important to treat like a conventional procurement process where it's just get 3 bids and make sure they're qualified and then pick the cheapest one or whatever, right? That's not going to be successful most of the time, in my experience. So understand the company, understand how they work. There are the big CMOs, right? They've got great track record, to Haro's point, great track record, but how easy are they to work with? How quickly are they going to just kick your slot down the calendar because of a problem with another customer, et cetera. And it's tough. CDMO business is hard, to be successful in, but it's how they partner with you that ultimately makes the difference, even when things are going well, but certainly when things are challenging.
I think one thing I can add, personal experience, in our clients, I tell them, I give them my cell phone as a CEO of the company. If you have a problem with my company and it's bothering you, you contact me. And I'll take care of it. That's very important. You won't get that from the big companies.
Can you tell there's passion around this question? I think all three have worked for CMOs and also hired CMOs, and it's a great question.
Yes.
I love that question, yes.
I think just echoing to what all of you have said, but I think it's not just the CMOs, right? Suppliers as well, right? Cell culture media and whatnot. You want to work with partners who understand your challenges, who really listens to your problems and give you as much attention, whether you're a small company or a large company, that they also give you that equal amount of attention and the flexibility as well, right? To work with you and to respond and to react. So I think definitely it comes down to the people. You need to be really comfortable with them and know that, they're your partners, right? Kind of like a marriage.
Very expensive divorce.
There you go. There you go.
I agree with that too.
Very, very much so. Yes, please.
Yeah, I'm loud, so I don't need the microphone. You can hear me, right?
Well, it's for the virtual. There's a virtual audience, so.
They need it. They need it so they can hear you.
We'll do them a favor, and we won't use them with my. A lot to talk about here on what just came up. CDMOs, and I work at one. Let me disclose that. But you are part of the therapeutic development process, whether you like it or not. You are not a robot. If you are, quit and go do something else and get the hell out of the way, okay? Track record, absolutely. Track record has a dark side. It ain't broke, we ain't gonna fix it. But it's not good enough. I just heard you guys telling us all, let's say we get a stage 4 CAR T therapy for triple-negative breast cancer that gets approved next week. We're screwed if it's autologous because the systems aren't in place and the demand is an avalanche. Sitting here and moaning about the problem without engaging in solutions actively now serves no purpose.
Great.
I'm a hard ass, I'm sorry. Because I've thought about it and I don't have a solution, so I'm hoping smarter people in the room will. Allogeneic, we wave that flag, I'm an allogeneic guy too. That'll save us eventually, but not soon enough unless something happens there. But the other part of the CDMO thing that you brought up that I loved is starting material quality. You are obliged on behalf of the patient and the client you serve as a CDMO to make sure the stuff that you get to work with is good stuff or it's garbage in, garbage out. And CDMOs right now do a lousy job on that aspect. They take things for granted. And that's a broad brush I'm painting with. There may be individual CDMOs that don't do it, but it's a problem. And I'll shut up now.
Yeah.
That's a very good point. I think
Great comments.
I think I tend to agree with everything that you said. Allogeneic is not there yet, but I can guarantee you that autologous cell therapy will be there forever.
Oh yeah.
It's not going to go away. It's not going to go away. So people, they claim...
2 million manufacturing runs. Each individual patient gets their own CMG campaign.
Right.
Uniquely each time. We'd like to say it's all the same. It's not.
Correct. Correct. I think there, again, I'm not as smart as you are, but I think there are many ways of addressing the concern. On the autologous side, for example, we are talking about the centralization of the manufacturing of the cell therapy products they put in the medical centers when the clinical trials are being done. Automation, closed systems. There are many companies that are developing stuff like that that can address some of the concerns that you have. But we're not there yet. I tend to agree with you. On the supply, raw material, absolutely. We got to do a better job in making sure that we have the reliable suppliers for manufacturing. And it's not only the suppliers, I think the equipment that we use, filters, everything that we use, single-use equipment that we, products that we use. So those are very important points that you made, and I tend to agree with you 100%.
QBD, sore point with me. Okay, we do it. We evaluate platforms, and the typical QBD is, does it make enough of the cells? Are they still alive? Do they have the right markers or purity content? Okay, good, great, let's go. That's a crock. What about functionality? What do you do to the cells when you thrash them on your platform to get your 95% pure, 95% viable cells? In terms of if you cryoprocess them and put them into a 14-day growth assay, what happens? Guess what? We did that with a canonical standard use across the industry platform versus a new different platform, and the differences were scary.
Yep.
And people don't look at that. because people are in a
That's
hurry.
you.
Yep.
That's a team question.
You were bringing up all the points. Let's be honest. We've been sitting in these meetings for 12 years now. We still haven't solved particles in our final product. We still haven't figured out our QC. We haven't figured out our assays. As you said, there's a lot of things that we haven't found to get to the next level. And we all have work to do, and we need to work together.
Absolutely. Thank you. Thank you so much for the comments. Unfortunately, we are out of time. I sense a very passionate and engaging audience here, so thank you so much for all the questions. I welcome you to reach out to these panels speakers and talk to them. And as Dave said, it takes a village, right? So we all got to work together, and we have work in front of us.
Thank you
so thank you so much.